Label the sample ID on the EP tube cap and apply a barcode on the tube wall for easy tracking of sample information later. Wipe down the workstation with disinfectant to ensure proper cleaning and sterilization. Thoroughly spray disinfectant on the surface of the cage before sampling. Prepare a clean cage, remove the water bottle, and open the lid, ready for the procedure. Soak the tweezers in disinfectant, then pick them up and wipe off any excess disinfectant with a sterilized tissue to prevent contamination of the fecal samples, which could affect microbiological test results. Gently hold the mouse with one hand, check its ID, gender, and appearance, and massage the abdomen to stimulate feces expulsion. Use tweezers to collect the feces and transfer it into the EP tube. After handling each cage of mice, return the tweezers to the disinfectant to avoid cross-contamination. Scan the barcode to input mouse information into the health inspection module, making it easier to track the sample ID and mouse details in later testing stages. Once sampling is complete, return the mouse cage to its original position on the cage rack. Generate and print the inspection form from the system, then pass it along with the samples to downstream testing. Cross-check the physical sample with the inspection form to ensure the sample and form details match.